Hello, my name is Dan Tempest, and today I'm going to talk to you about erythrocytes, blood types, and their impact on transfusions, as well as a journal article that looks into also transfusion reactions and maybe potentially masking the antigens that are on the outside of erythrocytes and allowing for better results in transfusions. So, first of all, we need to understand what is an erythrocyte. So, an erythrocyte is maybe more commonly known as a red blood cell, and these are going to specialize in gas exchange, so specifically oxygen and carbon dioxide. The term erythrocyte, erythro means red, and cyte means cell, so you can understand why it would be called a red blood cell. And they're really red because of the hemoglobin protein that contains iron. And so that is again why the blood is red, is because it contains a ton of these erythrocytes that have hemoglobin, which is red because of the iron. In shape, they are kind of similar to like a round donut where there's a depression in the middle, and you can really see this in the bottom right picture. If I take my pen here, and I probably shouldn't pick red. So we have an indentation here, and that allows for more surface area for oxygen to enter the cell membrane and bind with the iron that's on the hemoglobin. So interestingly, in red blood cells, there is no nucleus and there's no organelles either. That's interesting because no nucleus means also no DNA. So there's no protein synthesis, no gene expression going on within these red blood cells. No organelles are important because there's no mitochondria to do oxidative phosphorylation and create a lot of ATP. What they do use for energy is glycolysis. But if you think about it, their main function is to carry and transport oxygen throughout the body. So it makes sense that there's no mitochondria because in oxidative phosphorylation, we use oxygen. So we're not going to want the mitochondria to be using the oxygen that we're going to want to be transporting. So it does make sense if you notice, and again in this picture, there are no organelles and there is no nucleus within this type of cell. They are the most numerous blood cell in the body, numbering, depending on the individual, between 4 and 6 million. And that really depends on the size of the individual, their fitness level, and also their location. The higher up in the mountains and the higher above sea level you are, you tend to have more red blood cells. Again, they are concave in shape, and that allows for more surface area. And their lifespan is about 120 days. All right, I do want to look more into the hemoglobin protein. So I do have a picture of this upper right. Hemoglobin, there's 180 million hemoglobins in a red blood cell, cytosol. So it's found in the cytoplasm of the red blood cell. On this, if I look here, we have 4 globin proteins. So if I cut this into sections, those will be our 4 globins, and that's what's going to make up the protein. Each one of those is going to have a heme group, and on that heme group, there's going to be some iron. Again, that's where that red color comes from. Each hemoglobin can bind to 4 oxygen molecules, and that's 1 per iron or heme group. So again, here, if we look at the iron, there'd be 1, 2, 3, and 4. Okay. So oxygen is going to bind to that iron. So again, each hemoglobin as a whole is going to bind to 4 oxygen molecules in total. Carbon dioxide, on the other hand, will bind to the globin part of the protein. So that's more out in this area and over here and here. Okay. Now, this is a crazy statistic that I came across as I was investigating this. So if there's 280 million hemoglobins within a red blood cell, and each hemoglobin can bind to 4 oxygen, that's over 1 billion oxygen molecules per red blood cell. So considering there's now between 4 and 6 million red blood cells within a human, we have insane amounts of oxygen being transported throughout the body. So that's what an erythrocyte is. I will now clear the screen. All right. So now we have to talk about the different blood types. So what makes the blood types different is their surface carbohydrates. That is what determines them, and these surface carbohydrates are often called antigens. So if you look over in our table here on the right side, I'll grab my pen again. We can see on the bottom here, we have the different antigens, and that's really what determines our blood type. So if we look at the blood cells at the top, blood type A is going to have only A antigens. Blood type B, only B antigens. Blood type AB has both of them, and I'll talk about why that might be the case genetically here on the next coming slides. And blood type O has no antigens, neither A or B. So again, there's 4 blood types: A, B, AB, and O. And I do want to also point out on this slide, we have the antigens that are present, the antibodies that are present. So depending on the blood type, you have different antigens, and this is going to have an impact when I talk about transfusions later in this presentation. Blood type A, you'll notice, has anti-B antigens, so it's going to be not allow or destroy cells that have that B antigen. There's antibodies for A on the blood type B, and again, that's going to destroy any cell with A antigens. AB has no antibodies present. That's going to allow people with blood type A to be universal recipients. And then blood type O has both of them. So blood type O we'll learn is the universal donor, but they can only receive from blood type O. All right, so the genetics behind this. There's a couple pieces that are pretty important to point out. The first is that blood type is what we call multiple alleles. So this means that the blood type is determined by multiple alleles for the same gene. In this case, there's 4 possible phenotypes, as I mentioned on the last slide. That would be A, B, B, or O. And these letters are the individual's blood types, and they refer to the carbohydrates, as I mentioned, or the antigens found on the cell membrane or the surface of the cell membrane of the individual blood cells or erythrocytes. So blood type A has carbohydrate A only or antigen A only. B has B only. AB has both A and B, and blood type O has neither. So the other piece is that this is codominance. Okay, so it's codominance because when you have an individual that is AB, so here's our individual that is AB over in the right diagram. There's also a couple AB individuals over here in the Punnett square that we have. Okay, and when we have that, both antigens will show up. So it's codominance because both completely show up. It's not a blend, it's both completely showing up. It also is important to point out that A and B are codominant, but O is recessive. So you'll notice in this image, if you have one A allele and one O allele, we have that here, as well as here, you will be type A. The only way that you can get type O is down here if you have 2 O alleles. So if we look over the pedigree on the right, we have a type A individual who has an O allele as well, and a type B individual who also has an O allele. Those are the parents. They can give rise to any one of the 4 blood types. So we have a type A individual here. We have an AB, a B, type B individual, and then we have an O individual. So all 4 blood types are represented by these 4 offspring from that parent. So multiple alleles because there are really 3 alleles: A allele, the B allele, and the O allele. And then it's codominance because A and B are equally dominant. They can both show up. All right. The next piece I want to talk about, because Rh factor also plays a role here. So Rh is an antigen that was first discovered on the rhesus monkey, hence the name Rh factor. And it's a protein that's on the red blood cell surface of individuals who are Rh positive. So if we look at the image here, on top we have Rh positive individuals, and the black dots are symbolizing that antigen that is present on their surface. So those black dots, I'll circle one here, that's the Rh factor. Now you'll notice, again, just to reiterate kind of some points that I talked about before, if we look at the B individual, here we have only B antigens, that's the blue one. On the A individual, we have only A antigens. And on the AB, we have a B antigen and an A antigen. And notice there's no antigens on the O individual. Okay, so it is also important to point out that as far as Rh factor, if you are negative, that's the bottom 4 individuals here, or red blood cells here, they do not have that Rh factor on their plasma membranes. As far as percentages, about 85% of the world of humans are Rh positive, and only 15% are Rh negative. All right, so transfusion reactions. So this is really one of the many reasons why blood types is so important. A couple things to point off right off the bat. If we look at the donor column, which is on top here, you'll notice that type O negative is the universal donor. People with this blood type can give blood to literally anybody. It doesn't matter what the recipient's blood type is. The other thing to point out is that AB positive individuals can receive blood from anybody. Okay. Individuals who are negative for Rh cannot receive from Rh positive individuals. So you look again. So if I clear the screen quickly, if I have an Rh negative individual here, I have an A negative individual, you'll notice that they cannot receive blood from even like an A positive individual. You cannot receive blood from them. On the other hand, if you are positive for Rh factor, you can receive from Rh negative donors. So again, same type of concept. If I have A positive, they can receive from that A negative individual as well as O negative. And again, the reason for that is the surface receptors. So the reason is the body recognizes those antigens that are on the surface if they are not familiar with them. So for instance, my individuals with Rh negative, they're going to view those Rh positive antigens as a foreign cell, and those cells will be attacked by antibodies and then removed likely by phagocytic cells, and therefore that recipient will have a host versus graft scenario here, and they're going to destroy those cells. Very detrimental. Doesn't pay to attempt that. So we used to, before we learned a lot about this, different blood types, we used to have a lot of problems with blood transfusions. Now that we know this, we can be more educated. It's also why it's so important to give blood, because this transfusion does matter. But it's all because of these surface antigens that really make the difference. So that leads me into my journal. Before that, we have to talk about pregnancy. Rh factor can also have an impact on future pregnancies. So here I have an example in the image when a father who is Rh positive and a mother who is Rh negative have a child that is Rh positive. And when that happens during the delivery, the fetus antigens can enter the mother's blood, and therefore the mother will produce those anti-Rh antibodies. And the next time she has a baby, that can create an attack against those fetal red blood cells. So the solution that we have come up to with, and this was introduced in the 60s, was an Rh immunoglobulin immunization, essentially an injection, and that prevents that sensitivity to the mother so that she can have future Rh positive babies. Huge win for medical when this was introduced. All right, so just to give some background on my journal article, was titled Chemical Camouflage for Antigenic Determinants. Stealth erythrocytes is essentially what they attempted to create. So in most transfusions, simple blood typing, the ABO and the Rh factor is sufficient to identify appropriate donors. However, occasionally, appropriate donors for patients with rare red blood cell types cannot quickly be found, and that situation can become life-threatening. So what they were trying to do is they're trying to create red blood cells that essentially could evade detection from the antibodies and therefore still help the person that is receiving the transfusion. So they created a hypothesis that covalent binding of PEG, which is polyethylene glycol, to intact red blood cells might mask or camouflage red blood cell surface antigens and thereby permit the survival of the donated red blood cells. They had some experimental design where the human, the mouse, and the sheep red blood cells were derivatized with MPEG, which is methyl oxypolyethylene glycol, and the effects on the cell structure, function, antigens, and the survival was investigated. So to go through some of the data that they found, this data shows that the more MPEG resulted in less agglutinization, which is essentially clumping of the red blood cells from antibody binding. So if we look, when there was no, I got my pen, when there was no MPEG, no masking agent, in other words, you see quite a bit of agglutinization in that image, and therefore the transmittance went up. Okay, but if we look at the 6 milligrams, which was the most masking agent, you can see there was less agglutinization, so less antibody binding, which is positive. It's what they want. They wanted to evade that antibody binding. Okay, so the next image here, the graph on the left measures the amount of cells bound to antibodies. So again, the higher the PEG, the further right on the graph, the less antibodies that were bound. On the right, we have the number of red blood cells that were being ingested by phagocytes. So what happens when the antibodies bind to the antigen, they essentially are flagging those for destruction, and a phagocyte would come by and ingest them and destroy them. But again, as we expose them to more PEG, which would be more over here, less over here, what you're seeing is less and less cells getting ingested by those phagocytes, which is again good because that means less are being detected, which was the whole point and the whole goal behind this. So this last image that I have here just investigated a little bit on structure, because obviously you still want a red blood cells that's fully functional and structurally sound. And you can see that both of these images, both the altered ones, which are on B, and the unaltered ones look the same. Okay, so the results of their experiment, essentially, type A and B red blood cells in humans were modified with the MPEG, and they showed a decreased anti-A and B antibodies binding, so less antibodies were binding. and they were also structurally normal. There was also less agglutinization or clumping as a result. And their conclusion was that this procedure for the antigen camouflage may have significant potential for transfusion and transplant medicine. Here are my references, and I hope you enjoy learning more about erythrocytes.